Assalamu alaikum wa rahmatullah. Welcome back, everyone, in our last day of research methodology or your guide to research webinar. So we have discussed how we can prepare a research, how to critically appraise any research paper, and the different methods in collecting the data or sampling techniques. Today, we will continue our discussion with including topics regarding the research governance and the certain approvals that we will need to obtain before conducting any type of medical research. So one of the most integral parts in including or in having a clinical trial as a type of research is including the public in our study. And there are different forms for this inclusion that can range from simple observational or even engagement in the deep structures of the research process itself. So one of these methods is the public engagement. And it is defined that where the information and knowledge about the research is provided and disseminated with the public themselves. So these can include the science festivals that are open to the public with the data sets or even with discussion on the research topic, and can include participation or with the colleagues or member of the public in the finding of the research itself. And some of these initiatives for the public involvement or engagement is the NIHR Involve method in clinical trials. Another form of the inclusion of the public in the study is the public participation. In this type, the members of the public or the patients as a subject or participant in the research study, such as in regular clinical trials, in qualitative study, or even in observational study. So there is the public engagement and the participation with addition to the public involvement as well. All of these three different techniques can be followed when we are preparing a clinical trial. So the public involvement is that the public is actively involved in a specific research project or research in general that will help to shape the research itself. This is an example of this is that the research being carried out with or by the members of the public rather to them or for them. And the public engagement, as we have discussed, is providing and disseminating the information and the knowledge about the research finding with the public, where the public participation is that the members of the public take part in the study process itself. So there are a lot of terms that are used for the public involvement, such as collaboration, engagement, involvement, and all of which focus on including the public in the research. So one of these common terms is the co-producing of the research as much more deliberative process than that of the public involvement, and can actually require public members and professionals to be involved in an equal footing throughout every stage of the design and also of the delivery of the research. So the key principles include sharing the power, that the research is actually jointly owned and the people who work together can achieve a joint understanding in the research process. Also, we need to include all the perspective and skills in the process and making sure that the research team include all those who can actually make a contribution to the research itself. And the respecting and valuing of the knowledge of those who are working together is of paramount importance in this type of study. Also, building and maintaining relationship with trust and emphasis on these relationships is key to sharing the power. So why do we actually involve the public in the research? These can include the ethical right, the democratic right, the utilitarian argument, and in some cases, it is mandatory for certain funders and for the ethical committee approval, In addition to, it can avoid the production of waste research. Also, including the public can help in revealing subtle nuances and keep it real with identification of any unmet need. Also, it can improve the quality and the design of the research that we are conducted in general. An example of public involvement around the research cycle is the process or the cycle that we have discussed yesterday, and we can actually involve the public in every step of these processes. And in regard to the ethical approvals, it is only needed when the members of the public are assisting in undertaking or in analyzing the research, such as looking at the discussion data or talking to people who have similar condition, which may even remind them of the negative experience they had, causing any distress. Therefore, in this case, additional counseling and support can be needed, and the Research Ethical Committee will need to check this additional support if it was available. So in these circumstances, we will need an ethical research and governance approval to include them in our study. So who to involve and how to find them? We need first to consider the three following questions. The first one is that who our target population is for this certain research project, and also who are the right people to involve and who will provide any insight into this topic. In addition to where would we find those people so we can include them in our study. So to identify the right people to involve is of paramount importance in any research. So for example, if we wanted to include patients, we will seek to find patients in an outpatient clinic where there will be no ethical approval required, and through clinicians, patient organization, or charities. While if we wanted to include the public, we will search through social media or any involvement platform, or even in a community or organization setting. And if we want to involve a certain community, we'll look through, for example, the geographical community groups, the social network, or any religious or cultural group. So diversity is important to succeed in any research project. Therefore, choosing the public to involve is very important to have a meaningful result of our study. So after we include the public, we will need to recognize their contribution, as the recognition can bring value of the lived experience, and it can show appreciations with non-financial recognition also, such as training, lab tours, any type of recognition that the patient or the participant will appreciate. So an important point for consideration is the data protection. We need to keep the names and contact details of the member of the public secure in a secured folder that is locked with certain password. We should also communicate how we will be using the details for the patients and who will have access to them, for example. And we need to check our departmental data retention policy and our data protection coordination. There are, of course, some national standards for public involvement. These can include inclusive opportunities on equal basis for all members of the public, and working together closely with them to achieve the best possible result and support their learning process. with the communication or having a certain communication contact details and assessing that impact on their lives with following the governance guidelines. So people-focused research in the NHS, for example, cannot be delivered without the involvement of patients and the public. So no matter how complicated the research is or how brilliant the researchers are, the patient and the public will always offer a unique, invaluable insight that these we cannot actually see or observe as researchers. Another important part in the research process or the governance is the quality assurance and the quality control. We need to understand the quality in a clinical research and know the differences between them to understand the statutory inspections in any clinical research, including the community finding and their impact. So, quality in clinical research, we will be discussing some of the major topics such as what is quality and its systems. So, quality is the same as the scientific integrity. This will have a robust protocol that is accurate, reliable, and we need to follow at all steps. Also, it can have clear, specific aim with the scientific question, with well-written and no ambiguity in any data that we are including. Also, it can have comprehensive details and should meet the legal guidance and governance requirements. In addition, it should be funded, resourced, and also ensured with sensible recruitment of the participants. So also quality need to be a trustworthy procedure with standard operating procedure and reliable, well-maintained equipment. Also, it shouldn't have unnecessary risk to the participant with safe procedures, medicine safety, and reasonable burden who have trained staff and informed participant in terms of the patient information suite. So for the quality control, it is defined as the operational techniques and activities that is undertaken to verify the requirement for the quality of the trial-related activities that have been fulfilled. Also, quality control in practice can include the monitoring, the management oversight, steering committee, and the data safety monitoring committee. While the quality assurance is defined as all those planned and systematic actions that are established to ensure that the trial is performed and the data are generated, documented, and reported in compliance with the GCP. So quality assurance in practice will include the audits, the SOPs, the policies, templates, and any inspection being carried out. So quality assurance can make sure that quality control is in fact effective and actually achievable in fact. So according to the GCP requirement, the systems with procedures that assure the quality of every aspect of the trial should be implemented at every step of the process. And according to the legal requirement for IMB, the trials and the guidance for all other clinical research, it should include the quality assurance, which will help in minimizing the error and detect any mistake as early as possible. So, poor quality trial is considered a waste of time, money, and resources, and also have unnecessary burden to the participant. Therefore, it is regarded as unethical. And the responsibility for quality is the responsibility of the sponsor who is in charge of implementing and maintaining quality assurance and quality control system with written SOPs. And the sponsor should review systematically and will appoint monitors and audits and can even appoint a quality assurance manager or a governance lead. And in addition, they can delegate these actions. Therefore, everyone who is involved in the conduct of the clinical trial must take responsibility for their own work. Also, the quality is the responsibility of the staff, such as in the recruitment of a qualified, experienced staff and in having training procedures for them. And it can be included in the protocol process as the peer-reviewed expert input and the scientific input with the statisticians that are being included. Also, quality can be implemented in the procedures, such as in the protocol, the checklist, the template that were developed, and every step of the development procedures. Also, in the equipment calibration and maintenance, and the appropriate supplies, contract, and agreement. So the standard operating procedures should include a detailed written instructions to achieve uniformity of the performance of a specific function and should have a clearly written description of how a particular task is to be performed, by which person and who is responsible at what time and what place. So these can be used to train staff and can help to ensure the consistency, compliance, accountability, and efficiency of every step of the process. In addition to this, there are some typical sections such as including the standard operation procedure, including the cover page, the introduction, the purpose, the scope, and the responsibility with every specific information that is to be included. Also, there are numerous examples to be included for the standard operation procedures, and these can be found in these following links. In addition, in the quality assurance, these can include the audits, which is defined as a systematic and independent examination of the trial to evaluate that activities were conducted and the data were recorded, analyzed, and accurately reported according to the protocol and the sponsor SOPs, the GCP, and any applicable regulatory requirement, depending, of course, on the type of the study we are conducting. So in the quality assurance, audit is required as an independent snapshot examination by the trained auditor. And the processes, documentation, and data should all be edited by the investigator sites, the vendors, the laboratory, the key document, and even the systems that are being followed. So audit is considered a systematic independent review process and is against the standards of the GCP, the protocols, and the SOPs to ensure that every step is actually being followed. while monitoring is considered part of quality control and is defined as the act of overseeing the progress of the clinical trial and ensuring that it is conducted, recorded, and even reported in accordance with the protocol, the GCP, and the regulatory requirements. So the purpose of the monitoring is to ensure that the clinical trial results are reliable and the treatment are effective, which are having real unbiased results. So GCP inspection can be conducted through review of the document, the facilities, and the resources, and it can inspect the sponsor or the hosted organization, the trial itself, or even the service providers such as the labs. So these inspections can be part of the routine monitoring or even triggered due to some issue or MA driven by the monitoring committee. So some of the common findings in the non-commercial studies include the critical and the major inspecting according to a study in 2016 and 17, and they found that most of the issues were due to quality systems or even due to monitoring in the non-commercial studies. So what are the implications of non-compliance? These can include issues of finding at inspection, such as the impact on organization and their reputation, and it can be considered as a criminal offense, as starting to conduct, for example, trial without ethical approval or regulatory approval, or failing to notify the end of the trial, or any other serious breach in the data or in the clinical trial that is not reported. So some of these frequent inspection findings include, for example, IT system issues or insufficient sponsor oversight or insufficient primary investigator involvement or poor medical overseeing with lack of data or following the GCP practice. So GCP, which is the good clinical practice, is considered an international ethical and scientific quality standard for designing, conducting, recording, and even reporting the trials, which involve the participation of a human subject. These can help in providing assurance that the data and the reported results are credible and accurate, in addition to assurance that the rights and the confidentiality of the trial subject are actually protected. So the GCP was amended in 2016 and including information with consistency with the principle of the Declaration of Helsinki and having unified standards that provide mutual acceptance of the clinical trial data by the regulatory authority. And it had harmonization in 4 areas, including the safety, quality, efficacy, and multidisciplinary approaches. In addition to increasing the efficacy details from sections 16 to 13 in the GCP. So the International Conference on Harmonization, which is an organization of regulators and pharma industry to discuss these scientific and clinical aspects of product registration, can be a useful resources to follow or even to inspect. So a clinical research is defined as any research involving human subjects. These can include the clinical trial using a medical product, having questionnaires, interview study, or research on biological samples, or even including healthy volunteers and patients. So, a driver for these good clinical practice guidelines was the syphilis study that was conducted in the 1930s, over 40 years of follow-up period for patients with syphilis who were actually denied of access to these medications. This was followed by the Nuremberg Court in the 1950s, which included a voluntary consent of the subject, and results should be fruitful and not producible by any other means. So the trial design should be based on the result of these animal experiments, not immediately by human trials. And we should avoid any unnecessary suffering and injury to the participant. In addition to that, if the researcher believes death or even disabling injury can occur during the trial, then this research is not legitimate and shouldn't be conducted at all. In addition to this, the degree of risk may not exceed the benefit of the experiment, and every possible action must be taken to protect the subject from injury, disability, or death. Also, the experiment should be conducted by a scientifically qualified personnel, where the subject may terminate the trial at any stage for their free will. After this case came the thalidomide tragedy in the 1960s. These were originally described as painkiller and sedative, and had widely prescribed as antiemetic for morning sickness in pregnant women, and has resulted in the death of over 12,000 birth defects in 46 countries. In this case, animal testing for birth defects subsequently was introduced, and they did indeed find these negative impacts to develop. After that came the Declaration of Helsinki, which had about key changes in the GCP in 2008, including that underrepresented population, it should be provided with access to participation, and also inclusion of the standard information in the protocol with appropriately qualified individuals who are able to take consent and should therefore be included. Where in 2013, there was also some modification or changes. These included increasing the protection for the vulnerable participant and compensation for harm requirement with access to result and benefit post the study. So these are the processes that led to the development or improvement in the research process, all of which came from a negative experience that have been introduced as new guidelines for the improvement of any clinical trial. So the directive law and guidance can include many regulation authorities, which, for example, in the EU can include to replace all clinical trial across Europe in 2020 and increase the harmonization across the world in these recommendations with safety reporting and simplifying these requirements for the patient requirement. So the principles of the GCP included that clinical trials should be conducted in accordance with the principle of the Declaration of Helsinki and their regulations. Also, they anticipated the benefit for the individuals and the society that should outweigh the foreseeable risk and the inconvenience. Moreover, the rights, the safety, and the well-being of the subject should prevail over the interest of the science and the society at every stage, and any available non-clinical and clinical information on the IMB should be adequately supported and to propose these trial to be taking place in the first place. So the trial should also be scientifically sound and even described in a clear, detailed protocol, with the trial being conducted in compliance with this protocol which has the ethical committee approval and their favorable opinion. Also, the medical care and the decisions should always be the responsibility of the qualified physician or the person in charge. Moreover, each individual who is actually involved in the conducting of the clinical trial should be qualified by their education, training, and even experience to perform their specific task. Also, a freely given informed consent should be obtained from every subject before the trial participation. And all clinical trial information should be recorded, handled, and stored as to allow the accurate reporting, the interpretation, and also verification, irrespective of the type of the media that is actually used. And the confidentiality of these records must be protected in accordance with the regulation, and any investigational medical product should be manufactured, handled, and even stored in accordance with the GMP and used in the accordance with these approval protocols. And lastly, the systems that assure the quality of every aspect of the trial should be implemented, aspects of the trial that are even essential to ensure that human subjects are protected and reliability of these trials. Additional information or principles from the UK clinical trial regulation was added to the GCP in 2006, including the right of each subject to the physical and mental integrity that must be safeguarded at every step of the process. Also, the licensing authority approval that is required with the protocol that must define the inclusion and exclusion criteria, in addition to provisions that must be made for the insurance that cover the investigator and the sponsor liability. So, any investigation in human subjects other than the non-interventional trial that is intended to discover or to verify the clinical or pharmacological or any other effect in one or more of the medicinal product, or that is intended to identify any adverse reaction to one or more of such products, or to study the absorption, distribution, metabolism, and excretion of one or more of such products within the objective of these safety or efficacy should be followed at all steps. So we can follow these clinical trial medicinal product guidelines to ensure that our clinical research or trial is in accordance with the regulatory guidelines at every step of the process. So research governance is defined as systems or regulations that ensure a good practice and high quality at all stages of the research process. This will help in ensuring that the researchers do the right thing in the right way at the right time. So we should also note that new legalization can bring existing regulation and guidance on the clinical research into one document in general. So the purpose of these research governance are to ensure the dignity, rights, and well-being of the public, in addition to improving the research quality and preventing any poor performance, also to prevent or to help prevent the misconduct and fraud in the research process. And it can cover the entire research process from the design, management, undertaking, funding, or even participating in the research. In addition to this, the policy framework for health and social care was developed in 2013, and it focused on the patient, the service users, and the public. It included also safer, more efficient, or more effective treatment or care of the patient with other services that need to be developed and tested. So it can be helpful in applying to do research to be less barbaric and even for the industry to see the places which are usually very suitable for conducting clinical trial and research projects that are registered. So these policy framework for health and social care include 19 principles. And each of these principles can be detailed or regulated for the chief investigator, the research team, the student and the supervisor, the sponsor, and any person who is actually involved in the research process. So the GCP can cover every step of our research process, including the documentations, such as the protocol, the trial record, the approval, the recruitment, consent, safety, and every step actually of the process. And the safety reporting is considered a legal requirement. According to the Medicine for Human Use Clinical Trial Regulation, it is defined as an investigator shall report any serious adverse event immediately to the sponsor. So the sponsor, in response to this reporting, should keep detailed record of all adverse events relating to the clinical trial, which are actually reported to them by the investigator of that trial. Also, annual reports should be included and can include two types of reports that need to be submitted during the trial. These can include the annual progress report and the development safety update report. And at the end of every trial, there should be an end of study notification and final report within 12 months. So the annual progress report should be submitted on the anniversary of the original ethical approval and should be updated on the site information, recruitment, amendment, and any safety procedures. Also, there are some templates and guidance that we can follow with copy that need to be sent to the sponsors immediately. In regard to the developmental safety update report, these actually replaced the annual safety report and introduced to improve the harmonization across the research process with comprehensive annual safety review from the investigational companies or the sponsor who is responsible for delegating these process. In addition, there should be an end of study notification that is sent to the required authorities that must be sent within 90 days of the termination of the study, where a final study report must be sent out also within 1 year of the study notification. And a more important consideration is the archiving of every data that we have for our research process. These include keeping a log of all documents and where they will be located, ensuring that the data and any software is actually retrievable at any moment. In addition to labeling the medical notes that is to be retained with electronic record and software readers that need careful archiving so that we can easily access them at any point. So how long do we actually archive these data? This will depend upon our local requirement, but it should be at least 3 years after the last marketing authorization. And if the trial involved any children, we should archive them until the youngest one is actually 21. And if we had any advanced therapy trials, such as gene therapy, these need be archived for 25 years. So the safety reporting is considered a legal requirement and should be recorded or reported for any serious adverse event that immediately occurred to the sponsor. And the sponsor should keep detailed record of all adverse events relating to the trial, which are reported to them by the investigator of that trial. So the safety reporting help ensuring that the benefits of a new drug outweigh the risk of, or any possible side effect, and it is the responsibility of the sponsor. And in the non-commercial studies, the chief investigator usually take on this responsibility and can delegate it to other qualified personnel. And according to the clinical trial regulation, this defines the types of adverse events and will have timelines for these reportings. In addition to this, any adverse event is defined as any unwanted medical occurrence in the patient administered a medical product, which doesn't necessarily have to have a causal relationship with their treatment. So these can be any unfavorable sign, symptom, or disease that developed after the introduction of our trial product. So these should have be recorded on any adverse event form or even a log on the medical note and should be kept and sent to the sponsor or any regulatory authority. For the collection of these adverse events, this should include questioning the participant at each visit if they did in fact experience any side effect, in addition to assessing for any abnormal lab results or ECGs with medical notes and any admissions or visit to other clinics or any unexpected processes. So these can include diary entry and any other logs we can use as a source of information. While adverse reaction is any unwanted and unintended response to the medicinal product that is related to any dose administration. Where a serious adverse event is any serious event that caused or even result in death, is considered life-threatening or require hospitalization or prolongation of these existing hospitalization, or even if it can result in persistent or significant disability or incapacity. It can also lead to congenital abnormality or death and birth defect, or any medically important event. So the difference between serious or severe, as the severity describes the intensity, such as mild, moderate, or severe, such as severe headache, where the seriousness is can be regarded as severe as the guide for reporting, for example, some of the scenarios on these aspects. So, serious adverse event can be divided into serious or not serious. These can be as regarded as adverse event or adverse reaction to our medication. So, a serious adverse event is actually where a casual relationship between the medicinal product and the SAE is at least a reasonable possibility for the occurrence of this condition. So the casualty must be assessed by a medically qualified investigator to assess for any possible side effects. So in this diagram, we can follow it to assess the severity and the seriousness of any faced adverse event or reaction. So the foreseeable serious adverse events are often certain events that can be predicted and can even be defined in the protocol as not requiring immediate reporting, as there is a method in place that is set for us to follow and prevent the worsening of this condition. So it can be defined, as we said, in the protocol, and it can be disease-related or pre-existing condition-related, such as, for example, in the elderly or serious medical conditions, these can be predictable. So the expectedness of any condition is considered as if an SAR is suspected or confirmed with a nominated individual that need to be determined whether it was expected or actually not. Also, it needs to be appropriately trained, but not necessarily a medical intervention. And we can refer to the protocol and reference safety information to follow the guidelines on treating this condition. So if the case of a suspected unexpected serious adverse reaction, these can actually vary in the nature or the severity of which is not consistent with the applicable product information. So these must be reported immediately to the sponsor and the required committee within 7 days of a life-threatening condition, or 14 days if this was a serious unexpected adverse event. So an event which is not listed in the RSI or even is listed but has occurred at an increased frequency or severity or at a different specificity is expected or referred to as a serious unexpected adverse reaction. And this table can present some examples of these side effects. So RSI is the reference safety information, and it is a list of medical events which define which reaction are expected for implemented medical product. So it may be contained in the summary of the medical product characteristic or even in the investigator brochure sheet that was written before the start of the study. So how can we actually assess the expectedness of any adverse reaction? So a reaction is only considered expected if it is listed in the RSI and at the same specificity and severity as that was listed, where a reaction shouldn't be considered expected because of the patient's medical condition, but rather due to the side effect of the investigated medical product. So the safety reporting for any medication should include the summary of the medicinal product characteristic, describing the proportion and condition of use, and if a drug was used outside of the license, this will depend on the risk of the investigator brochure. Also describing the physical and chemical feature of this medication, and it can be produced by the pharma company that is developing the drug. So an investigational brochure will usually contain multiple lists of adverse reactions in different sections across the documents or even in the appendix. So the section which should be used as the RSI must be identified by clear title or even by their drug reaction that is expected. So in the investigational brochure, it should only contain the serious adverse reactions, but also the adverse reactions that are headed in the safety section, and should be including a table using the current term of the International Medical Directory for the regulatory activities. And if this is the source of the RSI, we can follow these guidelines that are available on the following website, which you will be handed in the PDF guideline. So this can include list of undesirable side effects or adverse reactions, and can also have information on the population, such as elderly or in children. Also is considered specific to a product that can vary with the formulation and can give frequency of expected event to be monitored. In addition to this, we should follow all adverse events and assess them, and we should follow until the resolution or the event is considered stable, and we should record or stop the date and outcome, and we should also record the season date of any serious event that has developed, with reporting that usually include other copies of the serious adverse event form with amendment that is to be added. In regard to the blinded studies and the randomized clinical trials, the blinding should usually be broken for any serious unexpected serious adverse reaction, and the unblinded individual is subject to the medication interaction only, and we should unblind as few people as possible within the team with the process that should be covered in the protocol or the SOP to follow specifically. So, in the commercial trials, the oversight of the adverse event is the sponsor responsibility, where in the academic sponsorships, the Data Safety Monitoring Committee is in charge of reviewing any new serious adverse reactions within 24 hours of their occurrence. So these must include independent members or experts to review this process to avoid any bias in the process. And a serious breach is defined as a breach which is likely to affect to a significant degree the safety or the physical or mental integrity of the subject of the trial, in addition to the scientific value of this trial. There are, of course, some regulations that should be followed in these cases, such as those stated by the Medicine for Human Use Clinical Trial Guidelines. So a serious breach in the safety is regarded as breach of the GCP or the protocol, leading to death or hospitalization of the patient, and the illegibility of the recruited personnel, and the implemented medical product, or even failure to report these side effects and having persistent use of the wrong patient info sheet with systematic errors in the data recording. Also, these might be identified by the monitoring or the auditor, the participant themselves, or even by the researcher. So the sponsor must report these side effects to the committee or the monitoring committee within 7 days of their occurrence. And in the case of an urgent safety measure, these can be implemented immediately without waiting for amendment or approval. So in this case, the responsible committee can be informed by phone, and then we should follow up in writing within 3 days of these implementations. So we can include what measures have been taken, their reason, and the amendment form or the covering letter. And in the case of a temporary halt of the trial, we need to inform the committee within 15 days immediately. Also, it should have substantial amendment from the stop of the trial and any other amendment from what we have restarted, or even an end of study form within 15 days of the decision should be emailed to the responsible committee immediately. So as we mentioned earlier, there are two types of annual report. We have discussed the annual progress report and the developmental safety update. There is also the other type, which is the end of trial notification. So the annual progress report is submitted to the responsible ethical committee with updates on the site information, the recruitment, amendment, and safety, and should be emailing a copy to the sponsor. So the Developmental Safety Update Report was introduced in 2011 to improve the harmonization of the research process across the globe. This can include comprehensive annual safety review of the investigational product with the sponsor who is responsible for delegating these tasks. So the safety reporting, we can follow in these cases specified template that can be found in this following website with safety reporting or serious breach of any adverse event. And in regard on what to include in these daily safety reporting, this can include an executive summary with these following 20 points that must include overall information on the safety. So an important part of our discussion will be the experimental design and informed consent that is obtained from the study participant. So according to the WHO, clinical trial is defined as any research study which prospectively assign human participant or group of humans to one or more health-related intervention to evaluate the effect on an health outcome. So these are prospective assignments that is the key issue in any clinical trial. So the clinical trial is considered a registry for any federal privately supported clinical trial that we need to follow for conducting our guidelines. So who can actually sponsor a clinical trial? It is sponsored or funded by a variety of organizations such as medical institutions, foundation, or voluntary groups. So the percentage of the commercial funding of the research process can account for up to 80%, while the non-commercial can be 20% only. And some of the classifications for clinical trials can include first, the phases of any clinical trial development. As we have touched on previously or briefly, the clinical drug development is often described as consisting of 4 phases. So it is important to recognize at the phase of the development that provide an inadequate basis for the qualification of any trial because of the type of these that can have even several phases depending on the studied intervention. So as we mentioned, we have 4 different types of clinical trial phases. In the phase 1, this is the human pharmacology, which will have a gross safety, pharmacokinetic, and any possible pharmacodynamic assessment. In addition to in phase 2, this will have therapeutic exploratory phase with more safety evidence, with early even evidence of the clinical efficacy of the medication, and can help in assessing the dose of the treatment. Where in phase 3, these will include therapeutic confirmatory studies with substantial evidence on the efficacy and the safety with the risk benefit analysis. Where in phase 4, this will include therapeutic use of the drug with the safety or efficacy in clinical practice immediately. So as we were saying, there are some different phases in any clinical trial, but we also need to understand why do we actually undertake any clinical trial. So the cost of a healthcare provision and medical services are considered high. Therefore, they need to ensure that the available resources are actually being used wisely and efficiently. And this has led to the development of the notion of an evidence-based medicine, which is defined as the selection of treatment and intervention based on the scientific evidence of the efficacy and the safety of that treatment. So a randomized controlled trial is considered to be having the highest quality of evidence according to the Cochrane collaboration that consider it to be the most valuable in provision of evidence. And according to the International Conference on Harmonization, many of the ideas and principles that we will discuss are contained in three important documents that were published by the ICH. So this published the guidelines for the design, conduct, analysis, and even reporting of the clinical trial, which will be used to support the formal registration of any new medication. And even though that not all clinical trials fall into this category, the ICH guidelines are considered a good source of reference for all the work on the clinical trials that have been conducted. And these ICH guidelines have three of the most valuable from a point of view of a statistician design and analysis, which are that statistical principle for clinical trial that are included in guidelines 9 and guideline 3 for the structure and content of the clinical study report and 6 for the good clinical practice criteria. Another major consideration in the clinical trials is the bias. So, a key objective of the clinical trial design and analysis is to avoid the inherent bias in any trial. So, bias is defined as any systematic tendency to distort or exaggerate the treatment effect. So, we want to avoid making any error in the conclusions about the effectiveness of a treatment. And also, we need to reassure that the trial is unlikely to have any inherent bias, either in the way that we collect the data or the way we are organize the trial and the way we even analyze these data. So we need to identify the patient population of interest as a key step in the trial design and analysis. So when we run a clinical trial, we are hoping to make the inference based on a small sample of subjects. This will be inference about the bigger, broader population subject who might have been treated according to the trial protocol. We should also select a sample of subjects from the population of interest, as we need to select this representative sample of the subject population in general. Afterward, we need to choose an appropriate control group, as when we investigate any intervention of some kind, we will usually need to make comparison about the outcome that is seen in this interventional group. So after choosing the control group and the participant, we will need to choose the trial design, as we will need to choose one that meets the objective of the trial and also fit into any logistical or practical constraint and avoid any bias as much as possible, with giving us efficient estimation of the effect on which we are interested in assessing. After that, we need to record the appropriate and the relevant data to ensure that we are recording all of the relevant treatment outcome data, and also that we recorded all other data that can have an impact on the analysis and also on the interpretation of the data outcome. After that, we need to identify any relevant hypotheses and statistical methods, as choosing an appropriate and clinically relevant hypothesis and choosing statistical methods with which to investigate these hypotheses is very important from the start of the study. After deciding the hypothesis, we need to analyze and report the results, as we need to produce all relevant data summaries and even analyze the report in a comprehensible way according to the standard of the reporting procedure. So, in any clinical trial of a new medical treatment, we are trying to draw a valid and reasonable conclusion about what would happen if the new treatment were widely used in a large population. But there are, of course, some limitation of resources, and practicality mean that we can only study and collect the data from a small sample of patients that is drawn from the larger population or the community. So inference from a sample to population is deciding on how to choose that sample from the larger population. So how we have two distinctive requirements in choosing that sample. The first is ensuring that our small sample of data is genuinely representative of the larger patient population, and two, to ensure that we use the sample in a way which help in avoiding error and any misinterpretation. So a representative sample from a statistical and drug research methodology guidelines, we need to have the following code to be followed for a valid statistical assessment of the efficacy and the safety of a study drug. It is important that a representative sample of a qualified patient be selected from the targeted patient population. So the first requirement in avoiding bias is to try to ensure that we have a sample of patients who are probably representative of the wider patient population that we are actually interested on. Also, it should clearly have details if the sample is not representative of the patient as a whole, and if the trial will be biased in respect of inference about that wider population. So there are many methods in which we can actually assess or have a sample. One of these methods will be the random sampling technique. So the ideal technique for drawing a representative sample from a large population is to draw a random sample. So in a random sampling, every patient in the population will have an equal chance of being selected for that sample. And the sample is selected using random process in general. But of course, random sampling is not always possible, but will have some logistically criteria that need to be followed. So organizationally, we have to restrict ourselves to patients in certain centers, for example, and to those patients who happen to be present for treatment during a specified time or during the course trial. So a key technique for ensuring the representativeness of the sample is lost to us in clinical trial designs due to practicality and also reasons that are more simplified. There are other practical options that we can actually follow. These can include the sample frame with random selection or the quota sampling technique. And if we are restricted to a set of subjects that are available at a certain center only, we should consider constructing a list of all possible relevant patients and selecting randomly from that list. For example, if we have a list of all patient hospital number, then we can select at random from that list. So this is considered better than just accepting the next available patient who attend the center in general. So an example of this is recruiting family physicians and patients for clinical trial from 24 postal areas that were constructed with computerized registry and then selecting at random from those patients. So to generate any sampling frame of a household with appropriate aged children for recruitment, for example, and gathering basic demographic information about these targeted population from a census brief can be a reasonable sampling method. And from a database of all those households that are included in the census, then a random list can be generated, including any children, for example, under the age of 10. While another example of the sampling methods is the quota sampling. So if we are even restricted in the random sampling that we just have mentioned, and these cannot be selected, then we should consider potentially important subject characteristics, such as a specific age group or specific gender or disease severity. This will help us in ensuring that the sample that's been selected has a representative form for each of the subject group. And even though that it is not random, this can go some way toward giving representativeness of the sample toward the bigger population. So an example of this in the quota sampling is epidemiological study assessing the dietary changes in the diet interventions. But in most cases, we can usually have convenient sampling method, and patients will be recruited into most clinical trial consisting of convenient samples. And we use those subjects who happen to be available at convenient place and convenient time. And typically, this will mean that we will select the next group of patients who attend the center for consultation, for example. So this is not entirely random method, but has the practical advantage of the simplicity and the convenience. So we will be taking a break now for Salat al-Maghrib, and then we will continue with discussing the sampling methods. Assalamu alaikum wa rahmatullah. Welcome back, everyone. So, as we were discussing the different sampling technique and method, we will now continue with the random sampling. So, random sampling is always regarded as the best technique possible. But is it actually always better? So, the reason that random sampling is considered to be the ideal is that random sampling from a population can guarantee that on average, the characteristic of that sample will actually match those of the broader population, meaning that random sampling can give us some reassurance about the representativeness of the sample, which other method actually doesn't. But since that random sampling isn't usually possible in all cases, we can recruit the sample that we assume with the subject are representative of the population. So we must always try to demonstrate that they are actually representative by a good, clear, and comprehensive documentation of any relevant characteristic of those subjects who are included in the sample. So the second possible sources of bias in our sample could be the way in which we use the sample in order to draw conclusion about the wider population. So we need to use our small sample, however it was selected in a scientific and unbiased method. And according to the ICH guidelines, the most important technique for avoiding the bias in any clinical trial are blinding and randomization. So these should be normal features of any controlled clinical trial and followed in that regard. So in some cases, randomization can introduce a deliberate eliminate of the chance into the assignment of the treatment to the subject, and it can provide a sound statistical basis for the evaluation of evidence and also tend to produce treatment groups in which the prognostic factors are actually balanced. So the balance of the baseline characteristic can be one of the randomization technique. So in this case, we can see that we have balanced the male percentage in both of the medication group and their blood pressure percentage as a baseline characteristic. And in this observational study, there was an imbalance at baseline characteristic, including, for example, the age, the surgical procedures or respiratory procedures that were actually conducted. So, how do we actually randomize? In its simplest form, it is just the process of choosing at random which treatment of each patient should be receiving, and in practice, this is done by any software. So it can also be done by generating a list of patient number and a parallel list of any randomly chosen treatment code. And as each patient is entered into the study, then they are given the next available patient number on the list and the next available treatment on the list for that patient. So the patients are distributed at random for each treatment arm. An example of a randomized list is the patient and the treatment group, and we can divide the treatment group as we like, depending, of course, on the patient that were included. So randomization may not work well in any small trial, but if the process is done entirely at random, then there is the possibility that the numbers of patients who are assigned to the treatment can become imbalanced. In addition to this, more patients may be assigned to treatment A than B, as in the previous example, and also in small trials, especially this imbalance can make the analysis difficult to interpret or make any logistical implication in regard to this. So according to the ICH guideline in randomization in block, there is an advantage to be gained by this randomization method, but the block length should be short enough to limit any possible imbalance and long enough to avoid any predictability in this process. So in a blocked randomization, the random assignment of the treatment is done within a specified block or a group of patients such that within each block there is an equal number of each treatment that have been assigned, but also in a random order within that block. So in this example, we have a block of 4. There are 2 treatments in this example, and if we assign a block in 4 that have 6 possible configuration, that we can then divide the patient in accordance to them. So if we had, for example, 40 patients recruited in a certain trial in these 2 treatment groups, then the first step is to randomly select 10 blocks from that list below and divide the patients in accordance to them. So we will choose any blocks that we would like, 10 because we have 40 patients and 4 groups in each block. And this is an example of how this will be achieved by choosing the patient number and the block, then the treatment assignment. So the details of the block length should not be in the protocol to avoid any breaking in the blind of that randomization. And this is limited to the possibility of the investigators being able to predict the next treatment assignment from the previous knowledge of these assignments. Also, the block length may be varied within a given list as to safeguard these information and avoid their leakage in any step of the clinical trial. So this general principle of structuring a randomized list in order to avoid any possible imbalance can be applied for any other reason. An example of this is in multi-center trial that is desirable that the number of the location of each treatment group should be roughly equal within each center. But it should also be simple randomization that may not achieve these more complicated methods in general. So in another method of randomization is stratifications, and we can, in this case, stratify by center, especially in the small trials. For example, generate a separate blocked randomization list for each center so that the balance within each center is maintained at least approximately to avoid any other issues of imbalance. So this is an example of how we can achieve this randomization method. So in randomization, we can, as we mentioned, have more balance at the baseline characteristic or of the prognostic variables that we are assessing. So the same principle can be applied to ensure that there is reasonable balance between the treatment group in respect to the important prognostic characteristic of those patients. So we can stratify by center and by any important prognostic variables, especially in small trials. So this is often resulting in several levels of stratifications within that trial. So this is another example of how we can stratify by center and also by the disease severity. And in this type of randomization, we should not over-stratify because it will lead to too many data that we are unable to analyze with sufficient reliability. So it is not a good idea to try to stratify using too many factors, as this can result in an overall poor treatment group balance, especially in the small trials. So we shouldn't have more than 3 to 4 factors and choose the ones that are of most important to us in terms of any possible influence on the outcome of that treatment. There are, of course, some alternative techniques such as minimizations. So the treatment allocation by minimization is based on a different principle from that of randomization. So the first participant is allocated at a treatment in random order. And for each subsequent participant, we can determine each treatment that would actually lead to a better balance between the groups. This is in terms of the variables of interest that we want to look at or examine. So in this example, there is an imbalance between the nutrition counseling and the behavioral counseling group. Therefore, to have more balance, we need to enroll the next patient, a female aged 52 that is non-smoker. Therefore, this will have more balance between our two studied groups. So we need to choose a method of measuring imbalance between the groups and any other various options that are possible, with some simple based on more complex statistical criteria as the rule of the previous simplification method. Also, the protocol should specify any method of measuring these imbalances. And we should always leave an element of chance that need to be included, such as assigning the next preferred subject to the group that with the more probability. And according to the ICH guidelines, a procedure by which one or more parties to the trial are kept unaware of the true treatment identity is required in the blinding or the masking of most cases. So blinding is intended to limit the occurrence of any conscious or unconscious bias in the conduct and interpretation of the clinical data. And there are many different options for blinding, as the knowledge of the treatment identity can influence the patient or the investigator response to treatment, or even their assessment of the response. So there are 3 options, which is that neither the patient nor the investigator knows which treatment has been assigned. Also, the second is that the investigator knows, but the patient doesn't, while the third that both of them actually know the treatment identification. So, in the double blinded study, both the subject and anyone involved in the treatment or the clinical evaluation of the subject remain unaware of the true treatment identity. So, double blinding is the optimal approach and what we hope to achieve in every clinical trial, but sometimes this cannot be done in every trial. So, double blinding can require standardization of the appearance of the treatment and also the details of administration of that treatment. And this can sometimes be complicated and may, in some instances, be logistically or practically impossible, therefore, cannot be implied. So, if this was the case, a single blinded study is the next best option to be followed. So, if the double blinded studies are not always feasible, if then we should consider the single blinded study in which the subject is unaware of the true treatment identity. And even in a single blinded study, it is important to try to avoid the bias in the treatment assignment and use a central randomization service with ensuring that the decision to enter the subject precedes the knowledge of the treatment identity to which that subject will be randomized at. So, if the investigator is unblinded, then we should try to have the primary endpoint assessed by an independent blinded assessor to avoid any bias in the analysis. So the patient can often still be blinded provided that their knowledge of the treatment characteristic is limited and unable to be identified. And for the third type, which is the non-blinded study or the open label study, in this case, both the patient and the investigators are aware of which treatment has been assigned to the patient. Also, this may influence both the patient response and the investigator assessment of their response. And if possible, we can use a completely objective measure of the response to avoid any bias. So the blind should only be broken down when the data are clean to an acceptable level of quality and any intentional or unintentional breaking of the blind before this should be reported to the responsible committee. So it is important to implement that the procedures which will clearly reveal any early breaking of the blind should be included. Where if the blind is broken at the center, then we can consider closing further recruitment at that center to avoid completely any possibility of bias. So in summary, there are three types of bias, which is selection bias, which is defined by drawing a reliable inference in clinical trial that we need to select a representative sample and use that sample in a way to avoid bias. There is also an issue with the location bias, and randomization to treatment regimen is the key tool to ensure balance across those treatment groups. Where in the assessment bias, blinding is the key tool and knowledge of the treatment allocation can influence the assessment of the patient to that treatment. So, an example of unusual blinded study is the adherence that is critical factor for achieving the molecular response in patients with chronic myeloid leukemia. So, in this study, they included 87 patients with chronic phases of CMLE that was treated with imabitine 400 for a median of 60 months. So, they had achieved complete cytogenetic response by adherence to the monitoring during 3 months period. In this case, the patients were told that their adherence was going to be monitored by counting the number of their tablets that were returned, but they were not told about the monitoring system in the bottle cap. So, in conclusion, the lack of adherence to the oral therapy for chronic disease is a well recognized problem. And we have confirmed this by this study that only 25% of the patients with a potentially fatal disease fail to adhere to their optimal dosing at a median of 5 years from their time of diagnosis. And depending upon the randomization and the blinding, the clinical trials are ranked in different hierarchies. The first most reliable type of studies is the randomized controlled trial, followed by other type of clinical trials and studies that are not considered clinical trials, such as observational studies or any case report study. So within these categories, there are various possible study types with no clear census in the order of their hierarchy. So in the type which are not clinical trials, these can include cross-sectional survey of subject disease status, for example, qualitative descriptive studies of the subject outcome, cohort studies, and case control studies. So these are typically considered observational study of what happened rather than prospectively assigning the intervention. Often these can include historical data of no prospective aspect and sometimes can only be qualitative studies. So example of studies that are not clinical trial are the concurrent cohort study where they followed up and compared the outcome of patients who received an intervention and those who didn't. So merely observations. Also, these can include historical cohort studies where variation on traditional cohort studies are being observed. So another example is case control studies, which is a type of observational studies in which two types of existing group differing in outcome are being identified and compared on the basis of some supposed causal attribute. So in the case control studies, these can have often identifying factors that may contribute to the medical condition. There is also the other type, which is controlled before and after studies. These have follow-up studies of participants who received an intervention and those who didn't. And this can be helpful in measuring the outcome variables both at baseline and after the intervention period. An example of this is the mortality assessment in relation to smoking by the British Doctor Study, a classic case control study example. In this study, they merely assessed the mortality rate and with the smoking status. So this was based barely on observation, no intervention was applied, therefore it is considered case control study of the observations that were followed over 40 years in general. So another example can include the case series studies, which are descriptions of a number of cases of an intervention and an outcome. These have no comparison with a control group. And the other type is the cross-sectional studies that have examinations of the relationships between the disease and any other variables of interest as they exist in a defined population at one particular time point. So an example of this is studies which are not clinical trials, the non-randomized trial, in which the subjects are prospectively assigned to an intervention, but not using a random allocation method. Where in the single-arm trials, subjects are all prospective assigned to a single intervention. And in the biomarker studies, the prospective random allocation to one or more intervention, but even the outcome in these cases is measured and not in a demonstrable way, such as health outcome or measurement of a biomarker. So randomization control trial are not the only mean of collecting evidence of higher value, but also have become recognized as the gold standard in these methods. So randomized is considered the gold aim, or what we hope for when we conduct a trial. And according to the ICH guidelines, the most common design for a confirmatory trial is the parallel group design, in which the subjects are randomized to one or even two or more groups. So an example of this, in the case that each group receive one regimen or one treatment, and they will receive, the subject will be recruited in a parallel into these various groups. So in this type of study, it is the simplest to analyze and interpret. Also, it is less likely to suffer from problems which may render the results unusable or even difficult to interpret. Also, it should be the preferred options for most trials. So in the crossover of the randomized clinical trial, this will help in reducing the patient numbers. In the parallel trials, it can sometimes require large number of patients, and it may be important or even essential in some cases to use alternative method which may reduce the required patient number. So the crossover studies usually reduce the numbers of subjects who are needed, and because each one acts as their own control and therefore have natural variability in response, which is reduced for the purpose of treatment comparison. And another important point to consider in this type is the washout period between the treatment. So the washout period means the time between the two treatments before the second one is administered to allow for the complete excretion of the first treatment to avoid any overlap of the symptoms. So the crossover studies have a number of problems that can be or can invalidate the result. These include the chief difficulty to carry a crossover of the treatment effect from one study period to the next. So this problem is especially acute in the two-period crossover designs because they can have carryover effect that cannot be distinguished statistically. And because the tests for the carryover generally have low power, therefore we cannot detect its true effect in general. So to avoid the carryover procedures, these need to ensure that the washout period is long enough in relation to the half-life of the medication. So the washout period should be long in relation to the half-life, and how long will depend, of course, on the medication and their indication. We shouldn't also forget about the duration of the pharmacodynamic effect in general to ensure that the medication is probably applied. So if we had 3 treatments, then we need to have 2 washout periods, depending, of course, on our assessment method. So the baseline measurement prior to each treatment are recommended for all crossover designs. An example of this, if we had 4 periods, washout period, and 4 treatment design. So every treatment appear once in each column and once in each row, therefore avoid any complications of the observed effect. Or we can even design in which each patient doesn't receive all the treatments, but rather receive 1 or 2 treatments of each group. Or we can even design a study in which the patient receives the same treatment more than once to detect the true effect. So we can even design a trial that each patient receive the same sequence of treatment all at once, therefore detecting any true signs or symptoms in all of the patients at once. So we can have many different randomization techniques, depending, of course, on our medication and the actual effect that we want to measure. So for the BERT data, these essentially have the same designs and we cannot have different groups in the same study. So a simple study of a prospective assignment of the subject to a single intervention with the conversion of the healthy outcome that is made before and after the treatment is considered is essentially a type of degenerate crossover design that we can include. So in the crossover complex design that need careful planning, as more complex the crossover design is, this can offer substantial advantage in terms of the different effects which can actually be estimated, but they would also need expert statistical input to design and even to analyze, as some of the allocation treatment sequence are considered more efficient than others. So the possible problems with the time effect of these studies include the suitability only for certain type of indication, as we need to ensure that the disease is being studied that is stable, chronic, and return to baseline state during the washout period. So this will have no long-term trend over the time in the disease severity. Also, further issues of the crossover trials often have to last longer than that of the parallel trials. So, which can increase even the possibility that the subject will drop out of the trial before completion and need to replace the withdrawn subject that may diminish the main advantage of the crossover, which is needing fewer subjects. So longer trials can even have a logistical problem with the implementation process. So after learning about all the different sampling technique and the randomizations method, we need to learn about choosing the appropriate control group and some of the notes on the endpoint analysis. So how can we choose the control group? This is according to the ICH guidelines, which include 10 guidelines. First of all, the choice of the control group is always a critical decision in designing the clinical trials, so that the choice can actually affect the inference that is drawn from the trial and the ethical acceptability of the trial to the degree to which the bias in the conducting and analyzing of the study can be minimized. So this also affects the types of the subject which can be recruited and the base of the recruitment, in addition to the type of the endpoint we are looking to investigate, with the public and scientific credibility of the result and the acceptability of the result regarding the authorities or the regulatory authorities in general. So there are different types of controls depending upon our study types. These can be placebo concurrent control or no treatment control, dose response control, or active control, or even external control. Or in some cases, we can have multiple control groups with different types. So in the no treatment group, this is subjects that are randomly assigned to the test treatment or to the no treatment study group. And the principal difference between this design and the placebo control is that the subject and the investigators are not blinded to the treatment assignment. And because of the advantage of the double blinding design, this design is likely to be needed and suitable only when it is actually difficult or impossible to conduct double blinding. So can only be reasonable with confidence that regarding to the endpoint, which is objective in the type of the measurements. Where the second type is the placebo control groups. In this case, the subject are randomly assigned to the test treatment or to an identical appearing of the treatment, which doesn't contain the test drug. So the treatment can be titrated to the effect of the tolerance or even given at one or more fixed doses. So such trials are almost always double-blinded, and the name of the control can suggest that the purpose of the control is for detecting the placebo effect. So it is not always having major benefit, but rather the placebo control design can allow us to conduct blinding and randomization with including a group that receive an errant treatment. It can also influence the inclusiveness of the spontaneous change or the natural history of the disease, with the subject or the investigator expectation and the effect being in the trial, or even using a therapy or subjective method can be included. So, for example, the use of a placebo is primarily a matter of blinding treatment allocation rather than any other possible or presumed effect. Another type of the control groups is the dose response control using more than one dose. So in this type, in a randomization fixed dose, the dose response trial are subject to randomization at every several fixed doses group. So the subject may either be placed on their fixed dose initially or to a raised dose gradually. But the intended conversion is actually between the groups or the final dose. So either the lowest dose function as the control group or can actually demonstrate the dose response relationship, providing the evidence of their efficacy. So another type is the active control or positive group. In this type of group, the subjects are randomly assigned to the test treatment or to the active treatment group. And in such trials, they are usually double-blinded, but this is not always possible. And in the case of many oncology trials, for example, they are considered difficult or even impossible to blind. This is due to the fact that different regimen or different route of administration is included. So the active control trials can have two distinct objectives with respect to showing efficacy. The first is to show the efficacy of the test treatment by showing it is good as the effective treatment, or to show that the efficacy of the superiority of the treatment we are testing to the regular or usual treatment. So this type is ethically necessary if an effective treatment do exist for a serious disease that may occasionally be ethically acceptable to omit for only a short period of mild disease cases. Where in another type, which is the external control group, in this type, it compares a group of subjects receiving the test or the treatment with a group of patients that is external to the study, rather than an internal control group consisting of patients from the same population. So the external control can be grouped of patients who are treated at an earlier time in historical control or to a group that were treated during the same time period, but rather in another setting. So the external control may be defined or non-defined, and we can use the non-defined group that is particularly containing many information, usually being considered uncontrolled. So an example of this is the historical control studies in which the survival of patients with chronic phase chronic myeloid leukemia were assessed after failure on the interferon alpha treatment. Therefore, they only assessed the patient condition after they were deceased. So this is an example of the illustrated data of the example we just mentioned. So there could be another type of control groups, which is multiple controlled, especially this is conducted in the placebo and active treatment group. So it has the advantage of using more than one kind of control in any single study and using both the active control and the placebo. So in a similar manner, the trials can use several doses of the test drug and several doses of an active control with or without placebo. So this is common in equivalence studies where the placebo included so that we can demonstrate the new treatment is better than the placebo, and that the active control included so we can demonstrate it is better than the placebo, and the active control and the test are actually equivalent in their effect. So choosing an endpoint in a clinical trial should be based on the clinical relevance, and a primary or a secondary endpoint should be specified in the clinical trial protocol with some flexibility that is permissible in the case of exploratory trials, but the pre-specification is essential in any confirmatory trials. We could also measure the endpoint at baseline if this was possible, and we can consider taking several such measurements to get a good average value in general. In addition to this, we can take baseline measurement in the crossover study before each study treatment is started. And to record the values of any relevant prognostic factors at baseline and allowing the assessment of baseline comparability with possible influence on the outcome is our main goal. And we shouldn't use any stat significant test to compare the patient groups at baseline with respect to the prognostic factors. This is meaningless in the case of randomized controlled trial and sometimes considered misleading in the prognostic factors that must be evaluated by including the prognostic factors in the statistical model analysis. Also, when we are planning a repeated measurement of an endpoint during the course of the trial, we need to consult the statisticians and also have the optimum number and timing of the repeated endpoint measurement depending on the expected trend over time. And we need to note that repeated measurement of an endpoint doesn't necessarily give the trial more power or even make the trial more informative. So an endpoint measured at a single clinically relevant time point can work as well as any other measurement that is included. An example of a retrospective study is the novel hemato-oncology fertility score tool to predict the survival in over 80s with multiple myeloma. So in this case, they identified the need to develop a simple-to-use hemato-oncology specific fertility assessment tool to guide the treatment decision, and included the patient according to the score tool that predicted the survival over 80s. So these results showed that the HOF score tool can be used to predict the outcome in patients with aged more than 80. So the advantage of this study was that they looked at unique patient age group and were not aware of the studies that have looked solely at this advantage age group. Another consideration in clinical trials is assessing the pharmacokinetic of the drug. This includes assessing the drug motion and the drug effect on the body, where the pharmacodynamic effect assess the drug power and also the drug effect on the body. So in any trial, we need to assess what actually happens to the drug when it is absorbed. So assessing the drug absorption, distribution, metabolism, and even the excretion of the drug to avoid the accumulation of any other side product or even toxic products. So we will start with defining some of the major terms that we'll be using. These include the absorption, which is the process affecting the movement of the drug before entering the systematic circulation. where distribution is defined as the dispersion of a drug throughout the fluid and the tissues of the body. And metabolism is the irreversible transformation of a parent compound into their daughter compounds. And excretion is defined as the removal of a drug from the body. In regard to the exposure, we need to know how much of the drug is reaching the site of action in the tissues, and we use the plasma level as a surrogate indicator of this exposure. So in assessing the effect of the drug, we will assess the Cmax, the Tmax, and the time post the dose with the upper action effect of the drug. So the Cmax is defined as the maximum plasma concentration that is achieved, and the Tmax is the time to the maximum plasma concentration, where the AUC is the area under the plasma time concentration. And the half-life of the drug is the time for the plasma concentration to decrease into half of that effect of the drug. So combining the measures of the biological effect, we can, depending on these data, identify the impact of this type of medication. So in a typical pharmacokinetic study protocol, this will include healthy volunteers with exceptions in encouraging studies or studies that we already know some data on the pharmacokinetic combined with the safety and measures of the biological effect. Also, in the inclusion of both genders in the study and the consented and screened by the medical exam and clinical test, including drug screening. Also, we can assess the fasted state of the patient or a single dose study of the standard volume of water or even of the blood taken through cannula that is designed in the time of the interval. So this is an example of collecting the data for certain medication, including the unit, the date and time of the application of the drug, and therefore analyzing these data for assessing the plasma time concentration curve for all of these general data. An example of this is the plasma concentration time curve for IV versus oral administration. So as we can see, the IV concentration is almost consistent through time, while the oral medication can have a higher impact at first, and then this will be decreased gradually. Where in multiple dosing studies, we need to attain a steady state by having a peak time that need to be assessed over the half-life of the drug. So this is another example of the pharmacokinetic profile for drug A, for example. We can see that it has a peak effect for the plasma concentration, and then it will be starting with the discretion. So what are some of the undesirable side effects of pharmacokinetic? This includes making sure that the subject is compliant. For example, if they meant to be starved overnight or they have other medication intake. Also, the labeling of the actual date and time as well as the subject number. and sampling from the correct site or following the standard operation procedures in sample preparation for the storage. So there are, of course, some factors affecting the pharmacokinetic of the medications. These include the physiochemical properties of the drug, the product formulation, administration route, and the patient intrinsic and extrinsic factors such as organ dysfunction, disease state, or any other medication intake, or even drug-food interactions. Another major consideration is the bioavailability of the medication, as some of the medication can be destroyed in the gut due to the pH, where others are not absorbed or even destroyed completely in the gut wall. Others will also be destroyed by the liver, then entered into the systematic circulation. Also, the effect of food can be assessed as the time after eating on the plasma level of the medication. So, in the food effect studies, this has the objective of evaluating the effect of food on the rate and extent of the drug absorption from a given formulation. So, the single dose crossover fit versus fasted in a healthy subject is the targeted population using the high strength of the drug product and having high fat diet, high caloric meal. Also, in the no effect, if there was 90% confidence intervals in the fed fasted state, we need to assess the Cmax and the AUC ratio within about 125% of the clinical significance. So this can be helpful in gaining information on the effect of the food bioavailability of the oral drug, also on the labeling instruction on whether to administer the drug on an empty stomach or even without regards to meal. So, for example, in the clinical trial phases, in each phase, we can assess different impact of these medications. These can include the pharmacokinetic or dynamics, and also the fasted versus fed state of any medication. Also, in the renal impairment study, there is a decision tree in which medication to include and on which side effects that we need to avoid. We're also in the hepatic impairment studies. There is a decision tree of the investigational agent, depending, of course, on the severity and on the dose of that medication. So some of the key messages is that pharmacokinetic can tell us about the exposure tissues to a drug and also inform us on the dosing of both the amount and the frequency. It also has four main components: with the pharmacokinetic studies involving the construction of a plasma concentration time curve following any drug administration. So after assessing the absorption of the drug, we need to assess its metabolism in the body. So the metabolism can be conducted either after passing the gastrointestinal tract and going to the portal vein through the liver, where it will excrete the drug into the portal circulation. And there are, of course, some enzymes that are in charge of the metabolization of the drug. In phase one, this uses oxidase to introduce or even unmask the Beller group or drugs, mainly through some specified enzyme. Where in phase 2 metabolism, this uses transferase to attach the small endogenous Beller molecule to the drug. And this can have some conjugation reactions and increase the water solubility of this type of medication. So the outcome of this is that active drug is converted to non-active, as most drug will occasionally generate toxic metabolites with paracetamol. It can also have a broad drug to active drug. This is in the case of codeine or converted to morphine, and also in the case of lipid-soluble to water-soluble medication that can help in the conversion to the water-soluble for excretion. There is also another rule to be followed, which is the zero order elimination. This is a constant amount of drug that is eliminated by per 1 minute or per time. So this is independent of the total drug concentration in the plasma. And this is considered as a rare, mostly occurring condition when the elimination system is saturated, such as in the case of using ethanol. So this is a consistent proportion of the drug that is eliminated per time and the proportion to the amount in the plasma in general. This is an example of this zero elimination order of the first dose after being metabolized. So the variation in metabolism depends on the first pass clearance, the drug interaction, the genetic polymorphism, and if the patient had any type of liver disease. So the first pass of clearance usually occur in the gastrointestinal tract. And the extent of the removal of the drug by the liver during its first pass in the portal blood is through the liver en route of the systematic circulation. And in regard to the high bioavailable drug, this will lead to low hepatic excretion ratio of the drug. This can have the effect that not much of the drug will be removed by the liver, and doubling or even tripling the amount removed by the liver can make no difference to the bioavailability of the drug at all. So, as the hepatic enzyme activity can be affected by the extracted first pass of the drug in the doubled or halved duration of the medication, will of course then affect the bioavailability of the drug in the clearance phase. So in the low bioavailable drug, this can have high hepatic extraction ratio, such as verapamil, which will have a minor amount that reaches the systematic circulation and will have big effect on the bioavailability of that medication. So the common drugs with a low bioavailability that are susceptible to the first pass drug interaction include this following list with the percentage of their bioavailability shown in the last column. So the metabolism can occur after the drug leaves the hepatic portal vein and then enter the systematic circulation. So in the inducer and inhibitors enzyme, this will have almost 60 different members of their metabolized enzyme and can be induced or even inhibited by medication with range of variability up to 400 folds and can have a major cause of the drug interaction in general in the body after being observed. So this includes a list of the inhibitors and the inducers enzyme or isoenzymes and their substrates with the inhibitors and induced effect in general in the body. This is yet another example of the increased drug concentration and lower metabolite level compared to the fit and fasting state. So in this therapeutic window of the drug, we will have the toxicity level and the subtherapeutic effect, which is an unobservable effect of the medication. And in this case, we would need to increase the dose. So an example of this is the effect of posthanin on the levels of the enzyme induction, measuring the mean plasma concentration time curve of this medication following its administration. So this will show us the time per hour after the drug is being observed in even a period of 16 days. And yet another major indication that we would need to assess is that the occurrence of any genetic polymorphisms in the drug absorption. So this is the case of having an inherited gene variants which impact on the enzyme expression or even the enzyme function. This would result in little or even no enzyme expression or even dysfunction of the enzyme effect at all. So this is an example of a genetic polymorphism due to the coding variant of this gene and having an altered protein structure, therefore altered protein function in general. An example of this is the medication that is only observed by one enzyme. Therefore, in population who have this enzyme deficiency, they are unable to clear this medication due to their genetic polymorphism and having severe toxicity side effects, such as the ones that were assessed in this phenotype study of the codeine effect. This is the pharmacokinetic and dynamic variance according to the warfarin dose of these two enzymes that are being tested. So we need to understand that most drugs are metabolized in the body, and we need to understand that metabolic pathway for a new drug is essential in the development phase of that drug. Also, the impact on the dose and the route of administration is of paramount importance to decide on the appropriate dosing. We need also to assess the drug metabolized by any single enzyme at a high risk for variable blood level due to the drug-to-drug interaction and any genetic polymorphisms. So we'll be taking a break now for Salat al-Isha, and then we will continue with the structure of the research paper as our final topic for today. Assalamu alaikum wa rahmatullah. Welcome back, everyone. So, for our final topic of discussion today is the structure of our research paper. So, we need to understand exactly what to include in any research paper after we have finished of our trial design and the inclusion, or we have reached the write-up phase. So, the first part of any research paper is the introduction. In the introduction, we will have the background. In this case, or example, is a brief background about coronary vascular disease. After that, we will include also a background about our planned intervention. And for example, this will be low carbohydrate diet. After that, we will have a section about the rationale for the study or the association between CVD and low carb diet. And at the final end of the introduction, we will need to include or write up a brief information about what we are going to do or our planned intervention. After writing the introduction comes the methodology section. In the method, we will just write up our recruitment method, our study design, and all of the required steps that we mentioned earlier on. And after the methodology section come the result or the figures. In the result, we need to include our analysis of the data and what we have found. So an example of this is writing up results in figures or tables. And this will be need to include the table name, and also at the bottom of the table, we need to include any abbreviation or significant information that we need to mention. Another example of figures is writing diagram or any other plotted histogram that we have conducted in using RB Spaces or R program. So even in these figures, we need to include the figure name, which include what we are doing and what they are measuring and what is each abbreviation represent or each line represent actually. And after the result section comes the discussion part, in which we discuss our finding in relation to the current literature or any previous studies that have conducted similar results on similar topics. So even in the discussions, there are some parts that we need to include, including, for example, a summary of the result, which will be a snapshot of our impact and any other trials with related data. So we need to understand what is the anatomy of any paper. We need to understand the underlying foundation to develop any skills and to have some of the identification for different parts of scientific research papers and being able to critique these scientific articles in a strategic manner, allowing us to assess our knowledge and awareness or understanding. So we need to understand that each paper will contain specified structure, which we can refer to as the anatomy of the paper. These will include the title, the authorships and contribution for each study, the abstract, the introduction, the materials and method, the figures, and any link with the result, the discussion, and also the writing and the critical appraisal, which is following any part of the discussion. So our first point to start in any research paper is the title. So an example of this, which we have just mentioned, the effect of low carbohydrate and low fat diet in a randomized trial. So after that, we need to indicate the authorship of the writer for the study. And in this example, this will be the study coordinators, including their authorships and their association or departments. So after that, we will include the contribution section in which we can acknowledge anyone who helped us in our research process, even in the analysis process or in the drafting or critical revision. So as we mentioned in the bullet points, after the introduction or after the titles come the abstract. In the abstract, we will have a brief information regarding the background of our studied study or any other relevant information. We will also need to include the objective, the design, and the setting or the methodology of the study and our applied intervention. All of these need to be mentioned very briefly. And after that, we need to mention our results and what we have found. And if we had any limitation in the study, this needs to be included in the abstract of our study. So any abstract will almost look like this. It's a brief information that is mostly gathered in one paragraph or one set of data. And after the paragraph or the introduction paragraph comes the the abstract paragraph I meant, comes the introduction. So in the introduction, we need, as we mentioned earlier, to include a brief background of our issue and of our intervention that we are going to apply. And after that comes the aim or objectives, the method, the result, and the conclusion. So each part in the abstract represents an important part of our summary for the research that we have conducted. So the same principle will apply to any paper, and we can use as a reference for more example, the data set on PubMed. So an important part of these research papers is the data protection legalization and their implication on medical research. So the good clinical practice is an international ethical and scientific quality standard for the designing, the conduction, the recording, and even reporting of trials which involve the participation of human subjects. It can help in providing assurance that the data and the reported results are actually credible and accurate, in addition to the assurance of the rights and the confidentiality of the trial subjects that are protected at every phase of our research process. So the GCP principles apply in all clinical trials information that need to be recorded, handled, and stored to allow its accurate reporting, interpretation, and verification, irrespective of the type of media that was used. So the confidentiality of the records must be protected in accordance with the regulation, and systems that assure the quality of every aspect of the trial should be implemented in every step of our research process. And essential documents are defined as those documents which individually and collectively permit the evaluation and the conduct of the trial and all the quality of the data produced. So these documents serve to demonstrate the compliance of the investigator, the sponsor, and also monitor the standard of the GCP and all applicable regulatory requirements which are needed for the research process. So the documentation can include the trial master file, which include documents of the trial and can actually tell us the full story of the trial. And this is regarded as the responsibility of the sponsor, but may also be delegated to the chief investigator. In addition to this, the investigator site file include documents of the trial at the specified site and is considered the responsibility of the primary investigator. And these TMF files, the trial master file, is incorporated much of the site file if the study is considered as a single site study. Therefore, we would only need to include one of them. In addition to this, the documentation can actually facilitate the smooth running of the trial or the study. Also, it can help to document that the trial is being run according to the ICH GCP requirement, and it can help in providing the basis for monitoring, audit, and also inspections of the study at all times. So, in according to the GCP, the trial master file shall consist of all essential documents which enable both the conduct of the clinical trial and also the quality of the data produced to be evaluated. So those documents shall show whatever the investigator and the sponsor have complied with the principle and the guidelines of the GCP, in addition to those with the other applicable requirements of the regulatory committees. So the TMF shall provide the basis for the audit and for the inspection by the competent authority that need to assess these issues. So there are some typical sections in the trial master files. These should include the protocol and association documents, the ethical approval, any other regulatory approval, and the contract for the employees or any contact details with blank copies of the forms and template that is being used, such as the patient information sheet. Also, the data collection, the case report forms, the test result, X-rays, or whatever type of data that we are gathering. In addition to the safety reporting and the IMB information, including the information brochure or any other details of the medication. This can also include the committee minutes and copy of reports for their meetings. So this section can actually, in addition to those, include a copy of the protocol and the associated document, copy of the ethics, the site research team details, and the delegation log in any certain condition. Also, the subject screening and enrollment logs, the completed consent forms that may be separated with the file notes, in addition to the data regarding any collected information, the safety reports, the IMBs, including the pharmacy details, and monitoring and audit report that has been conducted at any step of the research. There are, of course, some advantages of the electronic versus the paper collection method or documentations. and the balance is currently shifting toward the electronic method rather than having a lot of paperwork to be documented. So to represent the case now with documentation, we will have a lot of papers such as the paper sponsor files, the trial master file, and the site master file, any electronic information, or any hybrid paper, and all of which are actually being transferred into electronic files rather than paper-based one. So there are some requirements that we need to achieve in regard to the approvals that we must obtain. As according to the MHRA, the data should be attributable, meaning we can access them at any time. They need to be legible and permanent, meaning they shouldn't be lost and following the guidelines of the committees. They need to be computerized this, and they need to have original record or certified of a certificate of the true copy of the original paper. Also, they need to be accurate, and they need to have data governance measures that need to be complete, consistent, enduring, and also available at all time for any inspection that need to take place at any point of the research process. So as most of these papers are being transitioned into electronic forms, even the consent is currently being carried as an electronic-based one. And these must have processes to ensure that we can actually trust that the person who's signing the consent is who they say they are. And we need to trust that the signed consent form hasn't been altered since that last copy. Also, we need to trust that when the signature was applied, and we need to demonstrate that trust if it was required for any audit or monitoring committees. Also, we need to provide a copy of the signed consent form and either in a physical or electronic form to the participant whenever they need it or to have it as a reference for information. So according to the requirement of the MHRA, the data transfer and data migration must be in accordance with a planned manner and managed properly with systematic overseen and must be validated due to the risk of alteration or even the risk of being or having lost data during the process. And it must be tracked with audit trails and metadata to track any changes that has occurred to these files. So the data transfer is defined as the processes of transferring data between the different data storage types, format, or even computerized systems. For example, from a USB stick to a hard drive, these will need to be mentioned in the data log. Also, for the data migration, it is defined as the process of moving the data from one durable storage location to another. So this may actually include changing the format of the data, but not the content or the meaning, such as from a paper-based data into an electronic one. Another important consideration is to have a true copy. So a copy is irrespective of the type of the media used of the original record that has been verified. For example, by a dated signature or by the generation through a validated process. This will be done to have the same information, including the data that describe the content and the structure as the original paper does. So, a true copy of an original record may be retained in place of the original record if a documented system is taking place to verify that the record integrity of the copy is actually correct. So the organizations should consider any risk associated with the destruction of the original record at all. So the requirement should also include the validation, such as of a scanned document. These need to be checked to ensure that they are complete, accurate, and legible. And the finding should have, for example, if we had found a skewed image, this mean any data or writing in the margin that have been lost. And this can include only a one side of a double-sided document scanned, and for example, not the final copy, such as an interim copy. Also, if it was illegible, such as too light or too dark, that we cannot actually understand what is written inside. And if this copy was not logged, there is no assurance that the documents have not been lost. Therefore, we cannot regard these copies as true copy of the original paper. And in regard to the security and the confidentiality, this will need to have secure access with a password and trained staff with secure servers and backup copies for accounting for any missing data or any other issues with the saved ones. And issues regarding these matters, the security and the confidentiality, can include finding shared password and smart card, no written procedures or no testing at all, or even that the study team doesn't know where the server is or who maintains it at all. Therefore, we'll have a secure server issue or backup issue. In addition to this, we need to consider the data retention by ensuring the protection from any deliberate or inadvertent alteration or even loss. We need to also secure the controls that must be in place and should be validated where appropriate, in addition to having backup and any disaster recovery or testing record. Also, paper copy retention or destruction need to be monitored, with the archiving process having the same rule as paper apply and protect against damage, alteration, or loss. In addition to this, the software and hardware may need to be archived as well, and having a hybrid system of processes for full verification of the trial must be documented and available for check at any point in time. And issues regarding these findings can include problems with everything we just have mentioned, inability to demonstrate the BI oversight, or having confusing file structure or destruction of the source data, or even having incomplete printout of the quality control and assurance, or having uncontrolled data storage, editable data with no audit trails, or even inappropriate black and white scanning of the colored data. These can also include frequent scanning errors and subcontracted scanning with the GCB non-compliance copies. So the drug discovery and development process included many phases, as we have mentioned earlier on. These can include studies of disease mechanism, molecular studies, or even animal studies. In the animal studies, these can include knockout mice or antagonist methods, all of which will lead to target testing of the receptors or the transporter, for example, and will even lead to the drug being promoted with the safety testing and afterward having human studies with drug approval and registration. So for the research and development process, it usually takes 10 to even 15 years with about 800 million to develop one new medicine at all. So in the first 5 years, this includes the drug discovery period with testing of over 10,000 compounds. After that comes the preclinical period, which can last for 1.5 years with testing for 250 compounds. And after that comes the clinical trial phase, which will include 3 to 4 phases, as we mentioned earlier on, and this can last for even 6 years. And after that comes the FDA review or approval, which can take up to 2 years. And if it was approved, then this will move into the next phase, which is the post-marketing period. So why do many drugs or medication fail to obtain an FDA approval at the final step? So the toxicities issue is associated or related to about 50% of the reason that medication actually fail to obtain an approval. As the long-term safety is still sometimes unpredictable and can only be seen after a long exposure period. Bioavailability is considered the second most commonly reason for drug fails with the metabolism, as for example, the half-life cannot be predicted or the drug-to-drug interaction or due to even some metabolites of the medication. This will also be due to lack of efficacy in the clinical studies, which is the case in 33% of medications, as due to issues of the understanding of the pathophysiology that is faulty, so hard to predict the efficacy, or due to improvement in the understanding of the disease pathophysiology that would actually improve the success in the drug discovery by a large extent. So this simple diagram can show the reason for drug failures in phase 2 and 3. This study was conducted in 2013 until 2015. The reason were mostly due to the safety issues or the efficacy of the drug. And this was different in different setting procedure or different clinical research type. So in the early development set, this need to include the traditional development approach and the disease target approach. So the drugs for the common disease or the personalized medicine is not usually exclusive, but some medication or conditions such as cancer can clearly lend themselves to have a gene chip-based personalized approach, while others, such as essential hypertension, are likely to be treated in the future much as now, but with an individualized approach for those not responding to the standard therapy. Also, a closer association of the diagnosis and therapy is likely to happen across the board with the invented medications. This is an example of the modern approaches to the target identification and screening methods. There are, of course, some new approaches to the drugs for managing pain. These include improved formulation with controlled release of the oral morphine, for example, or flash release of sublingual phenytoline RX. This can also include improved kinetic or metabolism characteristic with certain medication, or even having a new chemical entity such as a refinement of a known mechanism of action, or having a novel pharmacological mechanism, or even novel mechanism derived from drug in an empirical use. Another method that is currently being tested is the ligand-gated ion channels, as many of the non-steroidal drugs are not fully established in this method, but in the developmental stage. Also, the voltage-gated ion channel method or the enzyme blockers, which have some inhibitors or the development of the path that will be long for this type of medication. Also, in the G protein coupled receptors as a targeted mechanism for receptor blockers, which failed to produce analgesia in human subjects, but these are still under consideration. Also, there is the growth and trophic factors, which have effective results in phase 2 studies, but the studies are currently being conducted in phase 3. So for the novel pharmacological target, the evidence for the substance B as a pain neurotransmitter is actually being currently carried out, with substance B is expressed in the primary sensory afferent. So the receptors of the NK are presented in the dorsal horn of the application of the SP, which will have neurons which are activated by the noxious stimuli. So a prolonged or intense stimulation to this will need to substance B injection in the inducement of the hyperallergia. So these will induce the NK receptors that are internalized in the dorsal horn and therefore having an antagonist effect, which are anticipated by the test sensitivity of the NSAIDs. This is more of an explanation for this type of intervention that is being carried out. So the first antagonist molecule were considered as analog for substance B itself, and were generally likely to have partial agonist properties. So these are also considered metabolically unstable molecule and not bioavailable. But some of them were considered as neurotoxic molecules. This can also lead to breakthrough in the early 90s of the Pfizer published structure on the first non-peptide substance, and together with the knowledge of the structure and of the cloned NK receptors, this will enable the construction of a reliable and predictive pharmacophore. This is a table for the species variant in the NK receptor pharmacology, which the drug companies are currently following in a range of anti-septation assay to compare their receptor action with the standard analgesic. So clinical trials with the MK or its prodrug had some post-operative dental pain with no analgesia, but ibuprofen was active. And in the case of migraine, this didn't relieve the headache or the associated symptoms, where in the post-therapeutic neuralgia, this had no analgesia. So this is a summary of the clinical trials with this receptor antagonist and their effect or outcome. And this is BIT studies with the same receptors, suggesting that it does have ineffective in pain trials. So as a conclusion of these studies from the clinical pain trials, they lack any clinical efficacy in the pain margin trials, but not due to the insufficient dose or the lack of brain ventilation. So this has antiemetic effect versus the lower dose than that of the ineffective analgesic studies. And this antiemetic action requires brain penetration. Therefore, these are not considered as analgesic in human subjects, but just in animal models. So why is there a mismatch between the animal and the human after conducting a successful trial in animal models and then failure in human studies? This will need or have an effect on the nociception versus the pain, as there is no exact animal model for testing human pain. Also, substance B is not the only substance released from the C fibers, and having multiple peptides for other transmitters, fiber acting in parallel will lead to this effect, for example, and species with differences in the function of the transmitters and receptors are more likely than that of those who doesn't. So will there be any primary afferent peptide antagonist to have analgesic properties in humans? This, for example, will include the vasodilator of cranial and coronary artery, but most abundantly will include the neuropeptide in the dorsal horn of the spinal cord, or even having elevated levels of the CGRP, but these are actually measured in jugular vein of the blood margin patients. So this will not be currently visible in the subunit combining in the alternative way to produce any different receptors, and these have been cloned and reassembled so they can be screened in the receptor blockers. Also, these can be considered as a potent injectable receptors antagonist that showed efficacy in phase 2 trials for some studies. So, in summary, there is a cause for optimism in the novel analgesic mechanism that will translate into a new drug, but the progress has been slower and also more expensive than that expected. Also, the base of the basic research and the rate of production of the new target continue to be high. And the target engagement evidence from the BIT and the biomarker facilities can actually have objection on these decisions. Also, novel drugs tend to fail in the late stages in the clinical development, regardless of their therapeutic area. And much of the emphasis is on the biomarker and the disease pathophysiology to facilitate the work on the currently intractable diseases and may not help with the common disease such as chronic pain. And for our final discussion, we will continue with discussing the final step in the research process, which is avoiding plagiarism and having reference. So first of all, plagiarism is defined as the presentation of another person's thoughts, words, or images and diagram as though they were your own. So you are also involved in these plagiarism complications that can occur with writing a research paper. And to avoid this issue, we need to have quote or paraphrase, or even have citation and reference list to comply with the good academic practice. So quoting is the case in which we use the exact words of an author in our assignment or research and adding quotations that will help support our argument and help to reinforce or even raise a new point. So this doesn't mean that we can just cut and paste any large section of text or insert quotation marks and hand in our work, but rather can be used in the case of a certain definition or quoting a significant finding in which we cannot paraphrase. So in the paraphrasing is the case that we read a piece of work and then rewrite it in our own words and interpreting these ideas of the original text for our reader. So paraphrasing demonstrates that we have understood the academic context of the piece and it allows us to support our argument. So it is not a simple word switching, but rather restating the word or the sentence in our own words. And the third most important step is citing and referencing anything we have included. So citing is defined as when we use another person's work, either by referring to their ideas or by including a direct quotation that we must acknowledge in our text of work. While a reference list is a list at the end of our work that we should include a reference to all of the sources information we have used. Where a bibliography that can include items which we have consulted for our work but not cited in specific, and these can be listed at the end of our assignment in the bibliography section. So there are some different forms for citing, but the two most popular forms are the Vancouver and the Harvard style. In the Vancouver style, each piece of work that we have cited in the text should have a unique number assigned in the order of citation. So if we cite a piece of work, then we need more than once, then we would use the same citation number for each time we mention the same result or same conclusions. An example of Vancouver style is, as you can see in this picture, how to include it in the text itself and how to write it down in the reference list. So in the reference list as a Vancouver style, we would list all the sources we have cited in our text, and the source in our reference list should be in the numerical order, and each number should match and refer to the citation in the main body of our text. So a useful method when citing or having a reference list is using the software or reference management softwares. These include or having the options of downloading reference from an online database, creating our own library of relevant reference, identifying and deleting any duplicated references, and organizing our data. This will also include sharing our library with the group, inserting a correctly formatting citation, and generating a reference list in a variety of output styles. And a popular program that we can use for referencing or citing is EndNote. So one of the features for EndNote is that it can automatically go and find full text of important information. We can search for and add citation info within the program itself. We can also share our library with a group or we can amend the basic Harvard and Vancouver style so as to fit with our regulations. And some of the other options that we can use, including reference work, Zetro, or Mendeley, all of which are regarded as useful reference method to be included. Thank you for your time. We have reached the end of our course. If you have any questions, please do send them on the chat so we can discuss them further. الله يعافيكم بالتوفيق جميعا. العفو بالتوفيق جميعا. بإذن الله راح نرسل لكم المحاضرات كاملة على الإيميل. بإذن الله أبشروا، إن شاء الله راح ترسل لكم نسخة من جميع المحاضرات. بالتوفيق للجميع. خلاص بكده انتهينا. اللي ما عنده أي استفسار أو سؤال يقدر يتفضل.